Welcome, everyone. Thank you for joining us for today's webinar, Automation Meets Cell Separation, Primary Cells for Drug Discovery Assays, presented by Lothar Ratti, Global Product Manager, Cell Separation, Miltenyi Biotech. My name is Xavier Gutierrez, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Miltenyi Biotech. For more information about our sponsor, please visit www.miltenyibiotech.com. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Lothar Ratti. I will now turn the presentation over to her.
Thank you also from my side, and thank you for joining our presentation today. Today we'll be talking about automation meets cell separation, primary cells for drug discovery assays. The agenda for today's presentation is, first of all, Miltenyi Biotech, the pioneer in cell research and cell therapy. We'll also look at density gradient centrifugation and cell separation directly from leukopak and buffycoat. We'll then take a quick step into automated cell isolation and finish up with the applications in drug discovery assays. So let's start. Miltenyi Biotech was actually founded in 1989 by Stefan Miltenyi. The basis of the max cell isolation is the column technology that he developed as part of his diploma thesis and patented in 1990. Already in the beginning stages, Stefan had a pioneering spirit, and he saw the potential for MAX technology to become a revolutionizer in the field of gene therapy. The CliniMax CD34 reagent system is a method for selecting hematopoietic stem cells from donor apheresis while depleting T cells that can cause graft versus host disease. The development of the system went on for over 20 years, and it included setting up a GMP manufacturing site in Teterow in Germany, and finally the CD34 isolation using the CliniMax together with the CliniMax CD34 reagent received FDA approval in 2014. Currently, the treatment of patients with acute myeloid leukemia is in first complete remission. What started out as one man's diploma thesis has since grown into a global company of 2,500 employees worldwide and over 20,000 scientific publications citing MAX technology. Annually, more than 2,500 patients receive cell products manufactured by Miltenyi Biotech Technologies. So what is MAX technology about? MAX technology requires only 3 easy steps. Firstly, you have the magnetic labeling, where an antibody conjugated to a tiny paramagnetic microbead binds to a receptor or protein on the cell surface. There are different strategies for doing this, and we'll get to that in a little bit later. The second step is the magnetic separation, where the cells are passed through a column that's positioned within a magnet. The cells labeled with the microbeads are retained in the column, while everything else is washed through in a gentle way through the column. The flow-through is called the negative fraction, and it contains the cells without the marker for the antibody or antibodies that were used for the isolation. In the elution step, the column is removed from the magnet, enabling the magnetically labeled cells to be washed out. This can be done either manually with a plunger to push the cells out, or automated using a vacuum to pull the cells out, or by increasing the flow speed of the buffer in the system. All of these 3 methods are gentle for the cells and separate the cells into the negative and positive fractions with exceptional purity and high viability. Therefore, MAX technology is based on three components. First, the MAX microbead. Secondly, the MAX column, and the MAX magnetic separators. There are several versions of each of these available, depending on your starting material, the cell type of interest, and whether you're using a manual or automated system. Since the column is the heart of the MAX technology, let's take a quick look at why we use it and why it's superior compared to column-free methods. The magnetic separators in a column-based and column-free systems have comparable magnetic force. The difference between the systems is the column that's filled with little iron spheres. Each iron sphere passes the magnetic force onto the next over a very small distance, which amplifies the magnetic force by 10,000-fold and ensures that the magnetic field is constant throughout the column. Thus, minimal labeling of cells with tiny nanobeads suffices to retain target cells in the column. The iron spheres have a cell-friendly coating, and the cells can run freely through the column and be washed thoroughly to remove all cell debris and contaminants. All this contributes to the fact that MAX columns allow for cell separation with minimal effects on cells. Therefore, MAX columns are the best choice for cell separation with minimal effect on cells for translational applications. This translational approach from benchtop to bedside is reflected in all of our products. We provide the same products in research grade as well as GMP and clinical quality to ensure smooth transition into the clinic. We provide the entire workflow of products for cell-based work, from tissue dissociation and homogenization to cell isolation and culture, including activation and expansion of cells, as well as cell analysis via flow cytometry in the end. Before we go into the specifics of primary cell isolation, let's consider the use of primary cells and cell lines in drug discovery. Primary cells are sourced by culturing cells derived directly from human or animal tissue. Cell lines, on the other hand, are generated by immortalizing primary cells with vectors and cultivating them over many passages. The cell cultures that result of this methodology, they differ vastly from with regards to resemblance to the tissue of origin and the characteristics and relevance to predict in vivo biology. So what are the differences? Well, primary cells generally are limited in supply because you have to have fresh material. Cell lines, on the other hand, are virtually unlimited because they can be cultured almost indefinitely. Primary cells have low proliferation in culture and therefore a limited lifespan. Cell lines, on the other hand, are easy to culture. And primary cells have high donor variation, of course, because all of us are different. All primary tissues are different. The cell lines, on the other hand, don't have donor variation. The cell lines that are derived from primary tissues have low mutation rates, whereas cell lines would have high mutation rates and may continue to change in culture. Primary cells are representative of the biological phenotype and the complexity of the biological system that they originate from. On the other hand, cell lines have low relevance in vivo, and they're focused. They need to be focused for research on a specific target. And finally, primary cells are pretty much pre-characterized and ready to use. And of course, the cell lines need to be immortalized first, and you need to have authentication. Of course, there are a lot of concerns over misidentified and cross-contaminated cell lines. For example, in a study of over 500 leukemia and lymphoma cell lines, 15% of the cell lines were misidentified. And also some argue that tumor cells obtained directly from patients are the best way to study cancer. These sources can be, you can ask me for the sources for these concerns later on. So primary cells are more relevant for the complexity of the biological system, the systems that we're working with. They can be a little bit more difficult to obtain. But how do we actually isolate primary cells? And primary cells provide more relevant data, which is, of course, what I just mentioned. So let's take a look at the use of primary cells in drug discovery and the steps involved, from your starting material to sample preparation, followed by magnetic cell isolation and the downstream applications. Most primary cells come from blood products, which are mainly human. There's a lot of flexibility in this area, depending on the number of cells you need, from whole blood and bone marrow with limited cell numbers to buffy coats and leukopads, which are larger in size and contain a lot of white blood cells. Tissue, on the other hand, can be of murine or human origin and can be either healthy tissue, such as spleen or lymph node, or tumor tissue. Patient-derived xenografts in mice are often also used in research. Whichever starting material you start with, in the past, they've all required sample preparation for you to be able to work with the cells. So let's take a look at the sample preparation for these materials. The sample preparation step is mostly density gradient centrifugation to get PBMCs, or in the case of tissue, dissociation of the tissue, for example, with the Gentle-Max dissociator, to get a single cell suspension. After this, you can proceed with the cell separation. We've seen these steps already previously: the magnetic labeling, magnetic separation, and the elution of labeled cells to get the population of interest. And depending on your project, you have different options to select from. Would you rather go for an easy and fast positive isolation, or do you need your epitopes free for a second sort? So do you prefer an untouched isolation where you can label everything else but the population of interest? Or do you have many samples and would rather separate them in parallel compared to one after the other? Would you rather freeze your aliquots of your PBMCs or isolate your cells directly? All of these factors are important in deciding which strategy is right for you. Once you have your population of interest, you can move on to the downstream application. Whether it's an additional separation step of subpopulations, for example, Tregs, or flow cytometry, such as immunophenotyping, we can recommend the kits that will make these steps easier. Or will you maybe culture the cells in vitro assays or run genomics or proteomics applications? Whether you're interested in maximum recovery, such as for cell culture assays, or high purity for reliable genomics or proteomics studies, we can help you with the strategy to get there. We'll discuss options of immunophenotyping and functional assays later. I'd like to take a look at the PBMC step before we move on to the examples though. As a summary of what we've learned so far, in traditional approaches, we go from blood product to PBMCs to target cell As a summary of what we've learned so far, in traditional approaches, we go from blood product to PBMCs to target cells in 2 steps. For this, we have the MAX microbeads for positive isolation and isolation kits for untouched isolation, where we label everything but the target cell. In this process, you have to go from sample to target cell via FICAL. I'm sure I don't have to tell you what FICAL looks like. Not going into detail, there are many steps. You have to centrifuge, you have to count cells. It takes a lot of time and it's prone to human error and user variation. Then this is why we're introducing the hassle-free alternative to Phycol, the straight from microbeads. The straight from product line allows you to isolate cells directly from blood products without density gradients, without cell counting, and in only a few simple steps. There's little hands-on, no user variation, and is the fastest available protocol for target cells from Buffycoat and Leukopak. The protocol is really simple. You transfer the Buffycoat, Leukopak, or LRSC into a suitable vessel and fill up to 80 milliliters if the volume is less than that. This example is for a Buffycoat, but it's similar if you're working with LRSCs or Leukopaks. Then you add the microbeads and incubate in the fridge, after which you can separate the cells using the Multimax cell separator. We'll get into the instruments in a little bit, but with this protocol, you can go from an entire Buffycoat to target cells in as little as 25 minutes, or from half a Leukopak to target cells in 40 minutes. Without density centrifugation, no lysis, and no counting. Now we can fill in the other option to Phycol, the straight from microbeads for direct isolation of target cells from blood products. We've just brought out the new products for Buffycoat, Leukopak, and LRSC. So to summarize so far, we've now learned about primary cells, what we need to consider when planning a project isolating cells, the steps to isolate them from tissue or blood products, as well as the shortcuts that we can take. Everything we've talked about aims at generating assay results that are biologically relevant, reproducible, and as efficient as possible, saving hands-on time and unnecessary steps and repeat work in the lab. But the reagents we work with are only one half of the solution. Truly efficient and reproducible results and protocols require automation. So I'd like to take a few moments to discuss automated solutions before going on to discuss drug discovery assays. So why do we need automation in the first place? As we've already said, automation reduces hands-on time and frees you to work on other more important tasks that cannot be automated. It minimizes variation between different users in the lab, or even good and bad days of an individual scientist. And of course, it minimizes costly human errors. With standard protocols and minimum user variation, you can be sure that the effects you're seeing are due to the assay. With standard protocols and minimum user variation, you can be sure that the effects you're seeing are due to the assay rather than artifacts. In short, automation reduces redundant experiments and allows you to focus your time where it matters. And very shortly, I'd like to introduce the Miltenyi Biotech automated solutions for magnetic cell separation. We'll start with the Automax Pro separator, the easy-to-use benchtop instrument for every lab. With the fully automated sequential separation from 1 to 6 samples, the Automax provides easy access to automation without high investment costs or lots of training. You can mix many different starting materials and samples within one run, and you can press the run button and the instrument will label and separate the cells for you. The columns are reusable, so the maintenance is easy, and there are preset programs for high recovery and high purity depending on your downstream application. If you have more than 6 samples or large samples such as Buffycoats, you might want to consider parallel cell isolation. The Multimax family is perfect for this. You have 24 magnets that allow you to process 1 to 24 samples in one run, or split your sample between multiple columns. This is achieved by using the LS, LZ, or whole blood columns, or the multi-24 column block for easy handling of 24 samples or an entire Leukopak. The Multimax Cell 24 is a semi-automated instrument, and it's a good fit for many labs that value easy handling but don't mind the additional pipetting. However, if you want full automation and maximum throughput, you will love the Multimax X, which integrates the Multimax into a liquid handler. It was only just launched 1.5 years ago, and this instrument is for the demanding high-throughput cell processing lab. Samples are labeled and isolated automatically, and you can go from an entire Buffycoat to target cells in 30 minutes or from an entire leukopak in 60 minutes. All programs are tailored to match your needs, so we don't leave the lab until you're satisfied with the protocol and the results. As you can see, the heart of the Multimax X is the Multimax Cell 24, with the elution chamber to elute the labeled cells and the deck layout around it that ensures fast and reliable processing. The storage hotels store deep well plates with buffer waste or the negative fraction, and the samples can be processed from 15 or 50 milliliter chill racks that keep the cells cold during incubation. The buffer troughs provide buffer for the isolation, and the 1 and 5 milliliter disposable tips are used to transfer reagents and samples throughout the deck. So in summary, we have the 3 automated solutions. The AutoMax Pro is great for small samples and low throughput with full automation. For example, when you're processing PBMC aliquots, mouse samples, or patient-derived xenografts. Because the AutoMax can process whole blood without FICL, it's also great for patient samples or for small donor samples. On the other hand, the Multimax family are the high throughput variants for fast parallel processing. For example, of small human or mouse samples, but also for large blood products such as Buffycoats and leukopaks. We've now learned that the steps of primary cell isolation and how to ensure maximum efficiency and reproducibility by selecting the best reagents and instruments to match your workflow. At this point, I'd like to take the chance to go through some application data from assays frequently run in drug discovery. And we'll start with phenotypic and high content screening. Phenotypic screening can be used for biologic and small molecule development. The example here is small molecules, where we looked at the binding of CCR2 on the surface of monocytes using high content screening in the development of an anti-inflammatory drug, a CCR2 inhibitor. CCR2 is expressed on monocytes, and it's the binding site for MCP1, a chemokine that attracts and activates monocytes. In the top row, you can see the blue monocytes on the left, the red MCP1 in the middle, and the overlay shows that the cells and MCP1 are co-localized. In the middle row, unlabeled MCP1 was added in excess to block all monocyte binding sites, and as you can see, no stained MCP1 can be detected. The same is seen when CCR2 inhibitor is added, so there is no MCP1 signal, and because there are no binding sites free. When you then look at the cells of interest, you can get much more information on a single cell level. For example, the cytoplasm shape, size, and area of the nucleus. The next example that we have is the use of cell separation in the development of mouse models. In this example of the humanization of a mouse, you would irradiate the mouse and transplant human CD34 positive hematopoietic stem cells, isolated, for example, on the AutoMax Pro. Infusing these cells into the irradiated mouse allows you to grow a humanized mouse immune system in the mouse. Of course, you can then monitor the success of the humanization by isolating the human immune cell subpopulations after the infusion. In the later stages of the drug development process, you'll be needing lots of primary cells for the mechanism of action profiling of your drug. In the example of an MLR assay, you will isolate monocytes from one donor and T cells from another to co-culture the matured DCs with the T cells. Adding, for example, a checkpoint inhibitor molecule allows you to track the T cell activation, for example, by interferon gamma production, as you can see here. The T cells and controls by themselves show no T cell activation, as you can see in the black, white, and gray bars. However, when you add the PD-1 antibody or PD-L1 antibody, you can show interferon gamma production in all three donor pairs as a sign of T cell activation in the orange and blue columns. Another method of MOA profiling is the Treg suppression assay. Here you isolate the T cells and stimulate them, and then add the Tregs. During the co-culture, you can track the inhibition of T cell activation by the Tregs. And adding the drug compound, you can detect the compounds that inhibit the Treg inhibitory effect, as you can see in this graph. The same method can also be used for example, the MDSC suppression assay, which is based on the same principles. I also wanted to quickly show you an example of a DCT assay for predictive immunogenicity. Here you isolate the monocytes and the T cells, add your peptide mix, and check which peptides cause CD4 T cell proliferation. These compounds have the potential to be immunogenic, and of course you need the response. Here to be as low as possible. And here on the right, you can see that the data correlates well with the clinical data that was gathered afterwards. The last example that I'd like to show is from the field of immunophenotyping. Here first, you would have syngeneic, for example, syngeneic tumors, but of course you can use patient-derived xenografts as well. You can dissociate the tumor, for example, using the GentleMax dissociator. Isolate the single cells to get the tumor cells and the tumor infiltrating cells, and then run immunophenotyping. If you look at the data without pre-enrichment, you can see that you can find hardly any cells because you're analyzing the bulk tumor. You're analyzing everything that was in the tumor. And if you only have a couple of cells in a gate, it's hardly scientifically relevant data. However, if you first isolate the TILs, the tumor-infiltrating lymphocytes, out of the bulk tumor tissue, and then go on to analyze those, you have a much higher frequency of cells that you're starting with. And you will get that relevant data that you're looking for, and you can be sure that the effects that you're actually seeing are the relevant ones that you're looking for. So in summary, we've learned today that primary cells are a valuable model for biologically relevant assay results. You should select the best to ensure maximum reproducibility, starting with single cell suspensions or going straight from to reduce the handling steps. And tailored automated cell isolation solutions make cell work more efficient and reduce handling errors in drug discovery assays. I hope I was able to give you some insight into the worlds of cell separation and their strengths and pitfalls to make your choice a little bit easier to select the best. If you have any questions, I'd love to get back to you. And thank you so much for taking the time to join us today.
Thank you, Laura Ratti, for that informative presentation. I would also like to thank Miltenyi Biotech for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May of 2019. As a final reminder, our speaker will follow up with any questions you submitted via email. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.